So we are here with our Microbiology Journal Club, our online journal club on Google Hangouts. And this is our first journal club of the fall, so I'm really happy to be back to this, reading some good microbiology papers. And then we've got a really great group today to talk about this paper, which is Growth Dynamics of Gut Microbiota in Health and Disease, inferred from single metagenomic samples. And I actually just read that in reverse because my screen is showing me like the mirror version, and I was like, oh God, that's in reverse. Am I going to be able to read all those? But I did.
Great skill.
So just to start out with, I think we'll just do brief introductions. My name's Laura Williams, and I'm an assistant professor at Providence College, which is a primarily undergraduate institution in Rhode Island. And I've started a small microbiome project this semester, so I'm very interested in how we can use different types of data to explore new questions about the microbiome. Since most of the time we're kind of just looking at who's there and then functional genes, this might be able to add another element to that analysis, so I'm interested in what they found in this paper. And Mark is right next to me in the little panel, so we'll ask Mark to go next.
Well, even though I can't get my logo up at present, I am indeed Mark Martin, your friendly neighborhood microbial supremacist, and I'm kind of the aged member of this wonderful triumvirate of speakers here. So I'm looking forward to learning what everyone thinks and communicating with the ideas. I tend to teach introductory cell and molecular biology in my beloved Matters Microbial in the classroom. And I'm doing my share of video casting. We'll see how well that looks. I must say, I have a perfect face for radio. Laura, you're great at this.
That's your YouTube thing. I really like that.
It's silly.
Okay, and so next to Mark we have Kat. Would you like to go next?
Hi, I'm Kat Milligan-Myrie. I'm at the University of Alaska Anchorage. I teach Introduction to Microbiology, and I'm interested in how the host has evolved to maintain individual microbial communities, and there are differences in immune responses to microbial communities, and I do this using the model Stickleback.
Great, thank you. Next to Kat, we've got Karen.
So my name is Karen. I'm a PhD student at Simon Fraser University, and my work is predominantly on a potential bioterrorism agent called Francisella tularensis, and I study its virulence genes and its interactions with the host. And so my interest in this paper is usually because I love microbes, and I like looking at the different questions that are involved with microbes, and definitely like getting various information across diverse fields. I love to hear about all those different types of information, and so that's my interest in the paper, but this will be wholly mostly out of my field.
Okay, and then Emily. Emily's next.
Hi, I'm a postdoc from the University of Alaska Anchorage, working with Kat, and I am brand new to the wonderful world of microbes. This has all been an exciting learning experience for me, and I'm interested in how host, environment, and ecology shape microbial community composition. Using 3-side Stickleback.
Excellent, thank you. So, and you, Emily and Kat are doing microbiome work now, yes, and so this is definitely up your alley in terms of using data, yeah. And last but not least, we have Dave and offspring, who's not currently visible.
Fitness. Gabe Ultras, University of Arizona. I do way too much. Mostly it's microbes interacting with other microbes, a little bit of microbiome stuff now, and usually in plants. I don't work with any mammalian things, but usually in plants.
Okay, great. So thanks everybody for being here today. I am really interested in what you think of this. So it's this is a science paper, which means that there's a lot packed into a little bit of space. And I will off the bat admit that I did not read the supplementary material because I just did not have time. So if somebody did, and at some point that becomes relevant, feel free to hop in because I have to admit that I didn't get to that. The main thing that they're doing with this paper is they're trying to take both metagenomic data that they generate and then also existing metagenomic data from database information, and they're looking to see if they can say something about bacterial growth dynamics. This is a really interesting idea because it would be very helpful to be able to mine our genomics data to say something about how different members of a microbial population are increasing or decreasing in their growth rate in response to different types of stressors or in response to different types of health states. Absolutely, this is a very appealing idea. And in the end, it becomes a very simple idea that they then test in various circumstances. And so I'm just going to show their little diagram here. You can see all my notes that I've made. So here's their little diagram where they try to explain basically what we're looking at is the difference between a growing bacterial population and a non-dividing bacterial population. The idea being that most bacteria have a single circular chromosome. There's one origin of replication, and then there's a bidirectional replication fork. So they, from that idea, they said, well, if we can look at the sequencing reads, if a bacterial population is actively dividing, we would expect to see more sequencing reads at the origin of replication than we would see at the terminus. So it's a relatively simple idea, and then they go through a lot of different conditions and different tests to try to figure out how, if you're adding levels of complexity, can you still see this pattern actually carry out? So I just wanted to, before we kind of launch into all of this, I just wanted to get a general sense of, as you're kind of approaching this, what did everybody think about in terms of the idea in general, and then how they start to go about it?
I'll jump in. It's a great idea in theory, and it works in a lot of cases. I had a particular, that one line in the first, like the introduction, where most bacteria have a single origin or replication, got me, because they cite a review, and that's kind of an old review, and there's a lot of other, like pseudomonads have multiple ones, and then there's all sorts of weird other stuff happening. And so it's not as easy as they say it is. And then so that's one thing, is that it'll work for some things, it may not work for other things, although it might just need tweaking. But again, if you're not culturing things and you're not doing the experiments in the lab, then it's hard to know how to calibrate that for microbe X in whatever environment you're looking at. Second thing is the assumption that growing populations are all that matters. And so I agree, it's awesome to be able to see who's growing and who's not, but there's a reason, a lot of virulence genes tend to be turned on in, what we call stationary phase, or places where growth slows down. So for realistic phenotypes, maybe, growth matters, but not growth matters too. So it's nice to be able to put things into different groups, but what that tells you is kind of hard to know yet. So.
How much of that disease angle do you think was, surely people are going to work towards that, but there's so many things that you could use this technique for that really don't have anything to do with disease. And I wonder if that's just like a hook you've got to get in there in order to make it really snappy, or do you think they really are chasing after, like, trying to, this is with Crohn's disease and this is with, diabetes, and we're going to be able to track, because they got to that towards the end, and that was a little more hand-wavy.
No, I think it's definitely useful information, and it'll tell you a lot, but it won't tell you everything, and kind of the devil's in the details with a lot of these things. So it's awesome that they can do it, and it's great that it works in these examples, but as always, like when you go to predictive models in places where you don't really know the biology yet, kind of hard to know what's going on. But that's that's not this paper. That's down the road for. I always pick fights with hyperbole and introductions with things. So.
Sure. Yeah, I think those are all really good points. One of the things you mentioned, I just made a note of, is they have a lot of, I don't know if verification is not the right word, but they did a lot of parallel results with doing CFU per mil counts, trying to look at abundance. So in this paper, when they're picked out some of the species that they wanted to work with, they have data tracking their metagenomics data. They've got abundance data tracking it. So I should probably back up here in case for people who might eventually watch this. The idea that they're looking at is this statistic that they're calculating is what they're calling the peak to trough ratio, and so that's just the idea that you've got at the origin of replication, if it's actively dividing, you're going to have a peak of sequence reads there, and your terminus is going to have like a fewer sequence reads there as well. So all their data basically comes down to this PTR number that they're comparing across different bacteria and across different communities, and that's that peak to trough. So if it's 2, I think they said that makes them conclude that it's an actively dividing population, and if it's around 1, they're concluding that it's not actively dividing. There's a lot in there. I mean, and they got a lot of data that's like, what does it mean if it's 1.4? What does it mean if it's like 1.7? And that I wasn't sure with that in terms of, when are you, is this something that you have to track over time, or can you take one time point and calculate this PTR and have it tell you something?
Well, I think there needs to be some, and they tried to do this, come up with some models where you can actually make predictions and test it this way. Mm-hmm. Because I'm, unlike you, more than a little innumerate. I mean, I married any mathematical ability that exists in the dyad at home. But what I would say is that I'm a little uncomfortable with them making those determinations until they get a prediction going on. And they try and do that with some of the bacteriostatic stuff. I was just teaching about gasp phenomena in stationary phase. And one would expect you'd pick stuff up that way. And Dave, did you see evidence of that in their data?
I didn't look closely enough.
Because it ought to be there, am I wrong?
In terms of what? The stationary?
You should see, like around RPOS, for example.
You're saying you should see, but is that expression or is that genome copy number?
Yeah, it's not copy number, so you wouldn't necessarily. I mean, that's going to change.
But then you are going to get like falling in among the ones that are pre-adapted to life in stationary phase. You should get an increase there. And so I guess this is something I was just thinking about as I was reading it, because I was just teaching about GASP, is shouldn't you see that there? Because out in stationary phase, you'll get a mutation that gives an advantage to some group of microbes, so they'll increase in number. And so you should get wave after wave. But I'm wondering if that's too small for them to see in their peak to trough.
Oh, so you're talking about GASP stuff.
I see.
Yeah, I thought, all right, I understand. The thing with GASP is that it cancels out, right? So
eventually, yeah.
Every division is met by a depth, basically. That's why they're stationary phase, and so I don't know that you'd actually see that. You could see, like, within a cell you might see it, but across the whole population I don't know that you'd see it because the number of chromosomes isn't necessarily changing.
Well, okay, leave that as it may. So can you think, Dave, of another test that you'd be happy with?
For growth or for
a proof of concept?
No, I think they do everything they need to do. I'm just not sure that I don't think it extends like they want it to extend. But that's everything. I mean, that's most things, right? So.
Right.
Yeah, I was kind of wondering with this too. There's a couple of like methodological things that I wondered about, and this gets back to me not having read the supplementary material, so this could be in there and I just didn't get to it. But does this? It seems to me like this would either require you to be able to assemble a pretty accurate draft of the genome sequence that you're interested in. So if you've got like this mixed microbial community, you either need to have a good quality reference genome to be aligning your reads to that you think is in that community, or you need to be able to do a de novo assembly to get full genome coverage of the species that you're interested in calculating this peak to trough ratio for. Because I think it's all just stacking reads onto a reference genome that comes from somewhere. So you're.
Yeah, I don't think you need a good. You can do it de novo. So that the challenge is getting long enough contigs around the origin and around the terminus, which should be. I mean, those are the DNA A, right? It's not. They're not like crazy duplicated, crazy repetitive things there, except for some of the smaller intergenic regions. So the power in this comes from, it's not the depth of reads at one position per se, but it's kind of repetitive depth across the origin, over thousands of kb.
Okay.
As long as you have, like, we tried this in the early days of like 2007 or so, trying to look for many of these draft genomes like duplications and plasmids, and
yeah,
the problem is copy number it's not normal, right? The Illumina reads are kind of biased, and they're still kind of biased in a little way, and for any position in the genome, if you just take coverage, it's not a nice, like, oh, here's 2 and here's 1. It's, there's a lot of slop in there.
Yeah, because there's still GC bias in Illumina data, right? That's not something they've addressed.
You need the window, and the longer the window you have around the origin and the terminus, then the better your confidence is. I need to go put on a new Mickey Mouse episode, so I'll be back.
Okay. Yeah, okay, so that makes sense. So you are looking at being able to reconstruct, as Dave was saying, like a piece of the genome around the origin, and then I assume a piece of the genome around the terminus. And if you can get that with confidence, then you can start to get that ratio calculated of coverage between those two. Okay, that makes sense. Yeah.
Well, what they're trying to do is really, really important to understand population dynamics remotely, right? I mean, that's the idea. If you think this whole concept of dysbiosis, which is more and more a part of trying to figure out pathogenicity and disease causation, I mean, it's really important. So I have a feeling that this work, I was looking at the authors, and they're mostly, number crunchers, looks like to me. And so, maybe this needs, like, other people to try it and expand it to outside so that they can bring more of a biological view to it. I mean, I know I'm kind of uncomfortable looking at how they're calculating things.
Yeah, I was just going to ask, actually I was going to ask Kat and Emily about that, because one of the things I noticed was they do mention that they had a pipeline they developed, and I think in the context of that pipeline there are a number of requirements or criteria that the data has to meet for them to use it to try to calculate this ratio. So there's a few times that they mention, we took this data set and there were only like 4 species that passed our filters that we could then calculate this for and draw some kind of conclusion. So I was just wondering if, like, you're thinking about something like the stickleback microbiome. I mean, if you wanted to use this technique, would it be a limitation that you put all this data through and then in the end you only get like 1 species that you can actually even calculate this ratio for? Would it, in a system that's expanding this to a system like stickleback, do you think it would need more tweaking?
I didn't look at their methods close enough to figure out if that would be necessary. I didn't see what they left out and what they left in. I'm kind of curious as to how applicable this is going to be to things like stickleback, where there's a number of different ways that you can use to measure the change in microbial populations, and I'm not sure that the change in the growth is really what I'm going to be interested in. I'm pretty happy with just taking a snapshot of what's there. This will be interesting, and the other thing is that one of the things that we can do with our stickleback is that the guts are transparent, so anything that, if we're really interested in looking at growth dynamics, one of the things that we can do is fluorescently label our bacteria and then actually follow the number of bacteria as they're replicating, because we can actually visualize them. So I think this is going to be much more interesting for models where you can't really visualize what's going on in the gut with the changes in the population. And I agree with Dave in that knowing which microbes are actively replicating might be interesting if you're looking at, like, how does the diet change which microbes are actively replicating or something. But once you start getting to what's going on in the gut in the overall population, I think that just following who's replicating and who's not replicating is going to be a small piece in a much bigger collection of data that you're going to be gathering.
Yeah, that makes sense.
I think So they do this in, the mammalian models,
Right.
which is great, but I think eventually we're going to be able to culture most of those things anyway. And so, especially with all the people working on it. So I don't know that this gives, because you have to, so to actually get the reads, you pretty much have to destroy the host anyway. So why not just culture the bugs and see who's increasing? Aside from that, I mean, the power with this is to go into like places where you can't culture, right? And so like Banfield's, Gillian Banfield's kind of acid mine tailings place where they know who's there, but they don't really have a good way to replicate that in the lab or actually let them grow really well.
Oh, interesting. Yeah.
So I think that's where you don't know what processes are going on and who's actually there and changing over time. I think it'll be more powerful there. And it's DNA is just DNA, and so it all kind of applies. I'm wondering though how long. So this is a great system. I'm wondering what the time frame is for how long it's useful, because we can already do, like in the lab, we can do things like ribosome pull downs now, where you pull down things that are actively replicating, right?
Right.
So there'll be much more targeted ways to get at who's growing and who's not in a much more specific way too. So with pulling down ribosomes, you can look for like which operons are the ribosomes attached to and where they're growing. So in Mark's case, maybe the gas stuff is happening. You can see certain transcripts being translated, even though there may not be a change in cell division numbers in the population. So that might give you more information. So,
and I think Dave made a really important point about things that are not growing that are having a huge effect on phenotype of the host.
Mm.
And that's something to be concerned about, of course. So, though I wish I could do a lot of the techniques Dave just talked about, pulling ribosomes out, but okay.
It's not easy.
Yeah, I was just going to ask. So here's a potentially naive question, because what if you're doing, like you said, if you're looking at operons and you're looking at what ribosomes are attached to which operons, does this give you more detailed information than just a transcriptomics approach, or different type of information than doing a sequencing transcript?
Oh yeah, so right, proteins are what actually matters for the cell for the most part. There's a lot of non-translated RNA too.
Yeah, fair enough. Yeah.
Transcriptome tells you a lot, but the proteins tell you much more relevant stuff.
Okay.
Yeah.
Yeah, that makes sense.
A lot of different ways to look at the same question give you different angles. So.
Yeah. Well, no, but when you mentioned the acid mine drainage sites, the work that's being done in as you said, the mammalian system is an obvious place to start, but there are some systems that are more difficult to deal with. Regardless of the technique, that you're right, that would be really interesting to be able to, because you could then follow that kind of longitudinally, and I assume one of the questions you could start to really get into is, are there kind of consortia, which I'm sure there are, consortia of microbes that one of them makes this product from that thing, and then that one uses it for another pathway to make this product that another one then uses. And I'm sure there are people who want to tease that out, and some type of approach like this would be interesting to be able to say, like, when the certain species are kind of increasing in number and decreasing, perhaps as a result of receiving some type of compounds that they can then use to metabolize.
That also gets eventually if the chromosome's getting copied, the rRNA is going to get copied too. So eventually you can just, count what we normally count, and you'll see, if you have the temporal sampling, you can see increases too. The utility of this approach is you see it in a snapshot, you see who's growing, right? Versus over time, there's different ways to do it.
Right. That's true. Okay. Yeah, that's interesting. Okay, so I was kind of curious about. So they did this. I was hoping some people could help me with this, because I was looking at their when they decided we want to see what happens. They did a lot of different tests, and one of them was to investigate the effect of an extreme dietary change on bacterial growth rates. 2 healthy human volunteers underwent an acute dietary change in which they shifted their normal diet to one that contained only boiled white rice for 1 week. At which point I wrote a little note that says, "Yikes." And Because I can't imagine if you just eat boiled white rice for 1 week. That's not going to be fun.
I think about grad school over there.
Yeah, there you go.
Except it would be top ramen noodles.
That's what they should have done. That would have been, every grad student would then read this paper and be like, what's happening to me? I want to know. But then they said their conclusion was, in both participants, which are probably two authors from this paper who decided to bite the bullet and do this, we observed a global change in gut bacterial growth dynamics between dietary regimens, as reflected in statistically significant differences in the peak-to-trough ratios of all bacteria between the days in which rice was consumed and the days in which the participants' regular diet was followed. And I kind of looked at the data that accompanied this, and I just kind of went, what? Like, I'm not entirely sure the best thing I can see is that you get a lot of variability among species. Is there something that I'm missing about this that they pulled out with this data in terms of dietary changes in the gut microbiome? I wasn't sure.
I agree.
Okay, good. It wasn't just me. I was like,
Are they actually quantifying negative change? Like, they say it's standardized, but how do they get a negative ratio? Is it from the, do they have a standardized? I'm not sure, so that's why I'm asking.
Yeah. Let's see. Shown are standardized peak-to-trough ratios and specific peak-to-trough ratios for species present in the sample. I'm not entirely sure how they did the standardization.
I'm not.
That might be another wade through the supplementary methods.
And it might be there, but
yeah.
It just seems like, that they are doing lots and lots of things here that are very different from one another. And I don't know about you, but I'm still bowled over by the white rice diet.
Yeah. Yeah, that's...
Why is this in the paper? That's the question,
It's in there because the reviewer said to do it. It's like...
Do you think so? Do you think that's it for this one?
I thought they were trying to show that this is applicable to changes in diet, and so one of the things you can do is you do this drastic, change in diet where you're going through from a high nutrient situation to extremely low nutrient situation, or at least a mononutrient system, and show that you can follow which microbes are actually responding to this diet. But it would seem to me that, I don't know, I had a hard time following this, their graph there and trying to figure out exactly what their major conclusions were from that. So.
Oh, good. I'm glad I'm not the only one, because I went through this and honestly, next to it, I don't know if you saw my comment, but I just wrote, not really sure about this. I just wasn't sure what I suppose if you,
The thing is, though, we can do this, this graph is superfluous because we have better techniques to measure those things over 7 days.
That's
the
what I'm saying.
one better.
Right.
But and isn't it kind of a silly? Well, silly. That's a strong word. Isn't it a non-optimal approach to use people for this kind of work because of the diversity of reactions and variability between individuals that we see because we're not genetically uniform?
Yeah, but this isn't cell-hosted microbe if they don't use people on that.
Okay.
Well, I was wondering if they were, if they have, if this is an experiment that they have going on for something else, and then this particular group is doing this part of the analysis for, this much larger experiment. I can see how, this experiment might be something that a group is doing to figure out how diet restriction is affecting the microbiota or something, and so they have another group that's doing 16S sequencing and monitoring health and monitoring the changes in metabolism or something, I don't know. And then this is something that they're doing with this same group. So it'd be interesting to see if another study came out using rice only versus normal diet in some other setting.
Yeah, that's a good point.
Well, from my perspective when I looked at it, I just thought that they came up with this technique where they had this theory and they understood that, okay, now we have this unique way of analyzing this data, and from what I was reading is it seemed like they just wanted to apply it to as many different types of microbial situations as possible, whereas like, oh, okay, look, you can use our technique when analyzing antibiotic resistance, you can use our technique to analyze virulence, you can use our technique to analyze abundance or change or diet changes over time instead of using cross-sectional data. That, I mean, and then they applied it to a general data set. So when I looked at it, it just seemed like we came up with this great method. Please use this across all different types of data as possible. That's just how I saw it.
I think that figure 3 is actually from reference 26, though, right? So I think they're using other people's data.
Oh, yeah.
That's what I'm saying.
Yeah.
That's probably just public data that they were using.
Yeah, but they're showing that, like as Karen's saying, they're showing like you can even mine existing data to try to put something together with this. Sorry?
To tell you what the other paper said already.
Yeah. Yeah, I mean, you know, like, because obviously, as you said, you can do, if you're just looking for who's there and how does that change, you can, as you've mentioned a couple times, there's other techniques to do that. But I mean, looking at this, you can kind of see there's some interesting, you know, I mean, just in terms of, for me, like thinking about what's happening in the gut, for instance, when you're on this rice-only diet, it is kind of interesting that between day 4 and day 5 and day 6, like you've got a couple of things that look like they hit a peak of, from this peak to trough ratio, what that indicates is that they're starting to divide actively, and then the next day they're dropping off. So, you know, that's just it gives you a good sense of how variable all of this is, how many different factors are in play, because even though these participants are on a rice-only diet, they're doing other things, there's other things affecting them, and so that could be playing into why you're seeing, in the 3 days that they were on rice-only, you're seeing some changes in terms of who's actively dividing based on this peak to trough ratio and who's not. Which is, you know, always something that people are thinking about when they're taking microbiome data longitudinally is, you know, what's changing and why, and what are all the factors that are affecting it besides just the one that we think we've manipulated.
Yeah, along those lines, I guess it would be, I guess where this would be applicable is where people are thinking about changes in microbial communities, and one of the things that they could consider is that the microbe abundance is changing because they're being wiped out by, you know, they're being killed by another species, or they're being killed by due to lack of nutrients or whatever, and so they want to see things, they want to identify things that are actively replicating as opposed to changing in abundance because they're actually, you know, being wiped out by whatever, by something else. So maybe that's where this is going to be more applicable when you're trying to determine if a population is changing in abundance relative to everybody else, or if they're actively dividing. I don't know. I'm just trying to think of other reasons that you would actually want to determine why, whether something is actively dividing versus just being metabolically more active, or just being, changing in abundance to everything else.
Mm-hmm. There's a, you know, we're very good at going out in the environment and sampling DNA now and kind of seeing who's there.
Mm-hmm.
I know a lot of people a lot of their research is focused on like who's there versus what's kind of remnant DNA that maybe not cells or maybe not. So I think this helps that, because if there's a difference between the peak and the trough, between the origin and the terminus, it does tell you that they're, you know, things are actively growing in the environment versus not there. Yeah, 2 computers. So, I mean, I know that a lot of people spend their research time trying to ask those questions. So, I mean, this will help that too.
Mm-hmm. Mm-hmm. Yeah, that's a good point. And I mean, so one of the things I was wondering about is, like, you know, so we've got this data. So that's the lactobacillus. I don't know.
What if you catch it here during sequencing?
Yeah, yeah. So right. So my question was basically like, what if you've got, so relative abundance is shown at the top graph, mm-hmm. and then you've got the PTR or the PT ratio shown at the bottom graph, and so we're looking at Gaceri basically, I think, tracks, let me make sure I get it right, it's the black one at the top. So
Right.
the PT ratio tracks here, which makes sense because it's increasing in abundance, so it must be actively replicating. And then it kind of starts to drop off because the idea is, I guess, you reach stationary phase, you're not actively replicating, and so the ratio is going to drop off. But so like if your data catches things here, it's going to be a low PT ratio, but you're high abundance. So I mean, how do you know where you are in terms of, because that part looks the same as the part where you start from. So how do you know where you are? You could have skipped the peak part and you wouldn't know. So I kind of wondered if that just requires more sampling, or if that just, if that's just a risk with coming in and taking samples at a certain time, is if you get a lower ratio, you can't really say anything about what happens before and what's going to happen. Because it could be that species is just about to start actively dividing and reaching towards a peak, or it could be that it's just come down off a peak and you really don't know.
Well, I guess we could look at the B. fragilis in that same graph and see that in the average CFUs, the number seems to be fairly low and actually decreasing, whereas the PTR is increasing in B. fragilis. So it makes it look like it's actively replicating, even though its abundance is still pretty low.
Yeah, I marked that, the green one, and I was like, like, what's going on?
Yeah. So maybe that's a case where you can say, okay, these are actively replicating, and pretty soon we're going to see those increasing, or you can start making predictions as, that L. gaceri has now used up the nutrients and is entering stationary phase, whereas B. fragilis is now able to survive off of either a byproduct of L. gaceri or some of these other products, or is now starting to, I wouldn't say thrive, but is now starting to become active again in the population.
Mm-hmm. Yeah,
so
that's
then,
a good point.
I was just going to say, so then the strength of the data comes because they have that relative abundance. Like they're analyzing over time, because then they can determine what the changes. But if you just look at the cross-sectional data from like Figure 4, where they just look at the patients, it's harder to, you get less out of the PTR ratios because you don't know what's happening before and after. Yeah, I agree with that.
Yeah, that's a good point. Yeah. It's interesting. Yeah. It's a really simple idea, but you can see kind of how it starts to get complicated as you're looking at all these different factors that get into it. Yeah. Very true. So I am kind of curious about, so Karen just mentioned this Figure 4, and they've got this, its bacterial dynamics correlate with several diseases and metabolic disorders. And so one of my questions with this is, I mean, how do you, I didn't catch this if there is, but like, what's your control for this? Like, how do you know stuff isn't just kind of, I mean, you've got these correlations with all of these diseases, but boy, I mean, obviously causation is not shown here, and that's why they're saying it correlates. But yeah. Would you use this and then, amongst other things, and then identify some candidate bacteria for different aspects of the progression of a disease state, and then try to tease that out? Is that kind of where they might go with this?
I could see looking at individual species, like there seems to be a fairly big change, or not big, but Bacteroides vulgata seems to change in, a certain subset of these different diseases and not the other ones. And so I could imagine using this type of information to say, okay, is this particular bacteria important for a type of disease that, something that's common between fasting serum, fasting blood, type 2 diabetes, whatnot, that's different from an inflammatory response like Crohn's disease or ulcerative colitis. These other ones, Crohn's disease and ulcerative colitis, are more inflammatory. I don't know, what was the BMI? I didn't see what that was. I'm not sure what the BMI stuff is, but it looks like there's like maybe bacteroides is not associated with inflammatory diseases. So if you were studying Crohn's disease and ulcerative colitis, you wouldn't put as much stock into how bacteroides is behaving in your population, maybe because it's more associated with changes in diet, or maybe you could start thinking about bacteroides being a species that actually responds to change in diet and change in insulin levels much more rapidly than other, or you couldn't even say rapid. It would just be changes.
Mm-hmm.
Responds to the changes in insulin, possibly, or something like that. I don't know. I guess you can start seeing from this. You can start seeing that specific species, specific members of the microbial community change in response to different diseases. But we already kind of knew that from 16S, so I'm not sure how this actively. Maybe I don't know.
Yeah, yeah. I mean, I'm wondering, they do a couple times, they make mention of that it looks like the PT ratio that they calculate is a predictor of changes in abundance.
Right.
So it comes first, which makes sense because, you've got to start, if you're going to be actively growing, you have to start by dividing, you have to start by replicating your DNA before you want to divide into two cells. That makes sense.
Mm-hmm.
I mean, I wonder if they're thinking about that predictive power in terms of, but it is a pretty small window, I think, that the PT ratio changes, and then not too long later, I think their abundance data reflects it. So I'm not sure. I'm not sure if we're to the point that having the information about what's changing that much faster is really. Yeah. Yeah, I mean, they looked at a lot of different correlations with a lot of different things that are certainly on people's minds in terms of what the role of the gut bacteria are. I mean, they have Crohn's disease and, as you mentioned, ulcerative colitis. Yeah, that'll be interesting to see how they follow up on that.
But I think the power really is that it's a snapshot, right? So you can take anybody's data, you can take it at any time. You don't need temporal sampling. You don't need accurate temporal sampling to make guesses as to who's going to increase or who's active in that environment.
So you don't think you do? You don't think, because that's what I was curious about with the idea that if you take on our temporal data, you end up like if a species is dividing and actively growing, it'll do a peak. But if you take your sample, if your sample comes from before or after that peak, you kind of miss that entirely. Or do you think that may not matter?
Well, so you're exactly right. But there's also a lot of people that aren't going to sample things exactly like your design may sample them, and if you're using other people's data, this gives you, you could design it so that you're sampling everything exactly like you would want to make some predictive thing out of it. But it's also possible that if you're just using other people's data, their experimental design isn't great, or there's only one sample, or you lose a sample at the key time. So it just gives you the opportunity just with one sample to kind of guess who's there. And yeah, there's going to be a lot of complexity in there.
Mm-hmm.
But on average, maybe it works, maybe it doesn't. But at least it's possible to do that,
Okay. Yep, that's interesting. Yeah, so I think this was it's interesting to see what people are doing with all this existing data, because we are just awash in metagenomics, genomics, 16S data, and trying to find different ways to tease out new information from it. As Dave was saying, that can be very valuable, because that data is sitting there, and it might as well get interrogated as best it can in all kinds of different ways. Yeah, very interesting. So does anybody have any kind of concluding thoughts about this paper? Do you see yourself, for people doing microbiome stuff, do you see yourself trying to take a stab at this? I know Kat already kind of said you've got better ways to do this, but do you see yourself kind of trying to take a stab at using this technique with your data? Dave, you said you were doing microbiome stuff. Are you off on a slightly different quest than this?
I mean, yes and no, right? So if we end up sequencing everything, then this will be useful at some level. I'm not sure when or where, but it could be, you know? So it's another tool, basically.
Yeah.
I'm going to change how I'm doing things because of it, but that doesn't mean I can't use it after the fact, too.
Yeah, that's true. I mean, and that's the point you've made that's valuable is that you can, the way they've done this, they've shown that you can apply it with existing data, and so you don't have to, as you said, it's somewhat robust to the experimental design, which is good.
I mean, if it works for just chromosomes, there's, you know, with more depth, you can probably tweak it for things like plasmids too, and actually get a feel for who's replicating in, you know, in reference to the chromosome, or if there's multiple replicons, like which ones are actually, you know, when those are replicating in relationship to one another. So it's, you know, just to have the statistics to be able to take a circular piece of DNA and compare one section versus the other. You know, you may not care about who's growing, but there's other questions you can apply them to as well. So.
That's a really good point about plasmids. Yeah, that did not occur to me, although it should have, because I think plasmids are really fascinating. But yeah, you could certainly do that with, and as you said, if you've got the chromosome there as well to use as kind of a baseline, you could then look at how different plasmids from their origin of replication, the difference between those. That's yeah, that's true. I'm kind of surprised they didn't try that.
I think this could also really help for people who are thinking about ecological differences. You know, how are the communities changing in response to, you know, the old standard is diet, but in response to stress, in response to light, in response to whatever whatever situation you want to see, that you want to actually measure in real time how the microbial communities are changing, and is it simply just a difference in the ratio of abundance, or is it that they're actively dying, or is it they're actively replicating? I think those types of questions will be, I think this type of information will be really helpful for those kinds of questions. So I think I'm not sure if it's so helpful in just, I have this disease, what microbial members are actively dividing. I think it's going to be more interesting for people who are thinking about the dynamics of the different microbes within the microbial community.
Yeah, that's a really good point too. Yeah, so there's some good extensions of this method that may not, in fact, follow the same track, but use, as both Dave and Kat have said, but use kind of the foundation of the statistical analysis and the methodology to ask some very different and interesting questions. That's a really good point. Very cool.
So
well,
there's also one, I mean, like always, there's a limitation too, right? Where if there's no diversity around the origin, if there's no diversity in the places you're mapping the reads to, then you're missing information as well. I don't know that that's that big of an issue, but you know, this isn't giving you the whole picture either, because within, you know, if the origin is pretty similar across strains, then you won't be able to differentiate those.
Yeah, that's true. I think I had, now that you say that, like, I think I did make a note of that somewhere where I was saying, well, what if you've got, yeah, you're right. So you could have an identical origin of replication, but you're amongst different strains, but their genome content is very, very different. And that could, you know, you could be like Pseudomonas, right? Like you could be looking at lots of different Pseudomonas doing lots of different things, but it would show up as, here's the origin of replication for this group of strains of Pseudomonas. Mm-hmm. Yeah, that's a good point. That is a limitation. All right. Well, I thought this was a really helpful discussion, in part because I had hit some parts of this paper where I was like, huh? So it was very helpful to get other people's perspective on it, and also in some cases the confirmation it wasn't just me. Does anybody have any kind of wrap-up thoughts? Mark or Karen, did you guys want to add anything before we kind of conclude here?
I feel stupid, so I need to work harder on this.
I thought this would be, I was pretty sure you might get into this because, like, as you've mentioned a few times, like, it's really important to kind of think about not only just the gut microbiome in terms of who's there, but how the dynamics are. So Mark, you've always emphasized that understanding the dynamics is important.
Well, I mean, it's very interesting to me, and this is related in a way. I mean, if you think about how we study microbiomes, we mostly look, please forgive me, at fecal material. So I've done some work where we're doing cloacal swabs, and they're not the same. And it's kind of interesting to think about that. And so at least with this technique, you're looking at like everything that's there and trying to look at the dynamics of change. I guess what I'm trying to do is to learn how to do the analysis.
Yeah. I mean, that's a learning curve for sure. But you took that course, so you're an expert now.
Oh yeah, absolutely. Absolutely. I can make a computer repeat something that I type in with the best of them right now.
That's a start, though.
I'm about ready to join one of the classes for 10-year-olds. I really am, because I think that would be a good place for me to start. Software carpentry.
Yes, absolutely. That's great. Karen, did you want to add anything before we conclude?
Well, I just thought that when I looked at the paper that it was a good way of just demonstrating a very simple concept, like coming up with a very simple concept, like thinking, okay, there's this origin of replication, and then you get multiple, because you get multiple copies, then you can get multiple reads, and from the fact that you get multiple reads, here's what you can do with that. And that was something that I really admired, because there's just so much it's very, very, very simple. It's a very simple idea, and then you can come up with so much more information from that very simple idea. And it's something that I'm trying to do. I haven't been able to do that yet, but maybe I'm not at the step of my career where I can do that. But I just admire that there's this depth of information that you can obtain from just a very simple concept. And so that was fun.
Yeah, I agree. And I think it's probably one of the reasons that I think all of us aspire to take an elegant concept and come up with something fabulous from it. But as I've mentioned a few times, I've not read the supplementary material, and I'm sure it's pages and pages. So what started out as a simple idea probably amounted to a tremendous amount of methodological sledgehammering to try to get to this.
26 pages in the supplement.
It's 26 pages?
26 pages.
Okay, well, there you are. So I do think they did a tremendous amount of work to try to get this to something that they could run all these different experiments, all these different conditions, and try to kind of take existing data and answer some of the questions we have about dynamics. Certainly not all of them, and there's definitely constraints, as we talked about. But as a couple people have mentioned, this is a good tool in a set of tools that we now have to try to interrogate some of this data that we have. So I think it'll be interesting to see, as Kat mentioned too, like if there's a follow-up to this, what they continue to do with it. Also,
I have one more thought that just hit me with that whole rice thing.
Yeah. Are you going to tell us how are you going to explain all of it here at the bitter end? You're going to be like, I figured it out.
Horribly flawed in more ways than one. So they're sampling out of poop, I'm guessing, right? Like that's got to be, if they're grad students, they're not sacrificing grad students.
Yeah. Well, yeah.
If they're measuring these temporal things, then that's totally off, because there's a lot of the microbes have traveled a lot since the gut environment, right? So, and at this level, the sensitivity might matter. So that's kind of nonsensical now that I think about it. It's only like if you're sampling in the environment that maybe you can know who's I guess what I'm saying is they're not measuring what they think they're measuring with those specific samples.
Yeah, that's true. That's another limitation. So there's an important point about, in terms of your experimental design, what are your samples, and how does that introduce a constraint to your data that you need to think about if you're going to use this method? That's true.
That might be why was it the bacillus that was off on that, or was I thinking of something else?
The bacillus, they had that. I think that was an in vitro community for the bacillus.
That's going to be, I mean, the power with this technique is you're sampling who's growing at over sensitive time frames, and so that's going to once you're sampling, that's going to be a different time frame than what you might actually care about with like human subjects. But
Yeah, true. Yeah, true. Especially if you're looking at if you're trying to make an argument that something is turning so quickly in terms of the way they said that the PT ratio predicts the abundance, but that was a short window. So if you're using fecal samples, that's going to adjust how quickly you can make a statement about something.
Mhm.
True. That's true. So some strengths, some limitations. Definitely an interesting method of tackling what exists in data sets that are publicly available and trying to interrogate them. So I'm really glad we read this paper. I think it was a good exercise for thinking about how to use your data. I am probably not going to try this with our microbiome project because I'm not going to try to get my undergraduates to to get through the 26 pages of supplementary material and understand the method. So I will just watch for others to use it. But, well, I want to, unless anybody has any final thoughts, anybody wants to try to take a stab at the rice thing again. But I want to thank everyone. Sorry, Kat?
I'm telling you, top ramen noodles would be much more real.
I really think they missed a trick there. They should have definitely done ramen noodles.
Would be selecting for halophiles by doing that, however.
Yes, that's true. That's true.
That's true. That's true. All right, well, thank you to everybody for coming to our first Micro Journal Club of the fall. We're going to try to do this once a month, and that depends on schedules, but I'll certainly let everybody know, and I'm always open for paper suggestions. So if you see something that you think is interesting that you'd like to do a journal club on because it gives you an excuse to read it, absolutely send it to me. We're pretty wide-ranging. I'm happy to kind of stretch myself to other fields that I'm not as familiar with. So if you're worried that it's outside the realm of what we've done before, please, I'm happy to take paper suggestions.
Thank you.
Thank you. Thanks to everybody. This was a really nice turnout. I was happy to have it for this paper. A good discussion.
Bye, Ernie. Or is that bird?
Bye, everybody.